everyone. This is Olga Redding with Iowa Organic Association. For those of you who don't know me, I am the Education Outreach Coordinator of IOA. Today we have a wonderful guest, Dr. Charles Benbrook, that will talk with us about the effects of dicamba and glyphosate on our health and our environment. So this is an amazing topic that we're definitely excited to hear about, very near and dear to all of our hearts, I'm sure. So what we'll do, just a few housekeeping things. What we'll do is initially, I'll go ahead and present just 5 slides about IOA and our mission, because we do post these recordings on our YouTube channel, and a lot of times new people view those. And then after my quick brief presentation that won't take more than 8 minutes or so, Dr. Benbrook will present. I will ask you to please mute yourself while the presentation is going on, and after the presentation, we will have a Q&A portion of the event. However, if there are questions that come up during the presentation, I welcome all of those questions to be added into the chat box. And usually I can go ahead and ask those questions of Dr. Benbrook at the end. And if you wish to unmute yourself at the end during the Q&A, you are welcome to do so. So just a little heads up, this meeting is going to be a little bit longer than an hour due to some new studies that were published and released recently that Dr. Benbrook would like to touch on. So we will plan to have about 15-20 minutes of Q&A that will most likely start around 12:55 or 1 o'clock. So just wanted to give you a heads up on that. And Dr. Benbrook, thank you so much for being here today and for presenting this very important, very critical information. We are thrilled to hear from you. And I'll go ahead and get started with my quick presentation. And thank you everybody for joining too. All right. So, Iowa Organic Association is a nonprofit organization that was established in the state of Iowa in 2006. And we are a nonprofit committed to organic education, advocacy, and community cooperation. Our mission is to advance organic agriculture and food systems in our state, and our members represent a diverse community of Iowa's organic farmers, food and farm businesses, advocates, and anyone that wants to support this very important movement. As far as our priorities, we have 4 overarching priorities to include education, outreach, advocacy, and building a community. So I'll touch on those really quickly. As far as education is concerned, we have been involved in various grants to include the Growing Organic Expertise workshops with technical service providers across the state. We actually delivered a training to NRCS leads last December, and this was a first of a kind training that was done in the state of Iowa. So we're very proud to have been able to accomplish that in partnership with Organic Agronomy Training Services. We also have been working with REAP-CEP, which is Resource Enhancement Conservation Education Program, and have been delivering growing organic expertise in colleges across the state of Iowa, and have been visited 17 colleges for the last 2 years. So that's been a wonderful way to be able to get in touch with students that will be the future farmers of America or in those fields. So happy to introduce them to organic. We also always participate in various conferences to include Marble Seed, O-Grain, PFI. So actually, hopefully you guys will be there at PFI this weekend. I'll be there Friday and Saturday. Look forward to seeing you guys. And then, of course, a big part of our outreach represents our website and our e-news, as well as our calendar that's always updated with events and information that is related to what's happening in the industry. As far as advocacy, we always continually meet with policy leaders to mobilize these very important organic interests in our state. We've met with Secretary Nag. We're now in the works with the TOP funding partnership, which has been announced recently, that will be about $300 million that will be funneled towards increasing programming for organic farming. And we have been selected as a partner for that. And then, of course, community, building community. This is what we're doing here today. So you guys are all part of this amazing community. So thank you for your support, and thanks for being here today. So, all right. And then as far as resources, the Organic Resource Directory is a document that we have available and update on yearly basis. It is available on our website through PDF form. So if you'd like to download that, feel free to do so. I should have some paper copies available at the PFI conference if you'd like to snag one. In addition, this webinar is made possible due to grant support from United States Department of Agriculture and Risk Management Agency, the RMA. And I just want to highlight some of the programs that are available through RMA that are available to organic farmers. So we have the Whole Farm Revenue Program that provides risk management safety net for all commodities on a farm under one insurance policy for any farm with up to $17 million in insured revenue. We have a micro farm program, which is similar to the Whole Farm Revenue Protection, but more geared towards smaller operations with up to $350,000 in revenue annually. And then they also have the Transitional Organic Grower Assistance Program that's geared towards producers who have crop insurance coverage on crops in transition to organic or certified organic grain and feed crops. And what's really exciting is we actually have a road show. We're working with the RMA next Tuesday in Davenport, Iowa. So if for any reason you're available to be there, it's going to be a workshop that one workshop will be geared towards agents in the morning, and then the other workshop will be geared towards producers in the afternoon. So be sure to check out our calendar listing and RSVP to that so that we know that you're coming. And then, of course, as an organization, a nonprofit organization, we rely heavily on your support. So if you're not already a member, please consider becoming one. It supports our mission, supports our education, and all of our efforts. So check out our website, and then feel free to connect with myself or Roz with more details and questions. And that is my presentation. So Dr. Benbrook, I'll go ahead and stop sharing, and then you can start. And Dr. Benbrook, I am unable to hear your audio.
That's because I'm not saying anything yet.
Oh, okay. There you go. There you go. That's why.
All right. Thank you, everyone. Delighted to be with you this morning. I'm going to work through a few slides to give you a sense of what we're up to and where some of the new data is coming from that I think is going to make a difference as the years go on in how farming systems in Iowa evolve. The reason that herbicides are so much in the news these days, and there's so much concern, is the obvious connection between GMO crops, glyphosate, and other herbicide-tolerant genetically engineered soybeans and corn in particular. When farmers rely predominantly on GMO seeds and herbicides for weed control. It increases the selection pressure on the weed populations, and that accelerates the evolution of weeds that are first less susceptible to the herbicide and ultimately resistant to it, which then requires the farmer to add additional herbicides into the mix, and away the herbicide treadmill goes. And the connection with organic farming is obvious. Organic farmers don't use herbicides. They rely on other practices to manage weeds. Roundup resistant weeds started showing up in Iowa in the around 2007, 2008. By 2010 or 12, it was pretty clear that farmers in Iowa were going to experience the same train wreck that farmers in the Southeast had experienced about 5 years before, as difficult to control, economically damaging weeds gained resistance to glyphosate. The response in the response by the seed, herbicide, and biotechnology industry that within 15 years or so ago, they really became one industry, was to develop new corn, soybean, and cotton varieties that were tolerant of multiple herbicides, in particular, two older phenoxy herbicides that would pick up some of the more damaging glyphosate-resistant broadleaf weeds. The first one to hit the market was Dicamba, the so-called Extend or Extendimax technology. There was limited illegal use in 2016, and then considerable legal use starting in 2017. And as you can see from this graph, the Dicamba use on Extendimax soybeans really accounts for the lion's share of total national Dicamba use. And the increase in cotton and corn is also meaningful. So when people back around 2015 recognized that herbicide use in the Midwest was going to follow a similar trajectory as it did in the Southeast as glyphosate-resistant weeds took hold and spread, a number of people were concerned about whether the rising use in herbicides would contribute to more serious or more frequent reproductive problems. And so I was asked by some folks at Organic Valley to what sort of study could be done to determine whether the increasing herbicide use in the Midwest was in fact associated with reproductive problems, difficult pregnancies, or adverse trends in children's development. And I said, well, there should be a birth cohort study done in the Midwest to track what might actually be occurring. And the team of scientists that came together to start the Heartland Study about 5 years ago, we certainly hoped that we would not find any association, but we felt that the science really needed to get done. So we started what's now called the Heartland Study. It's a large birth cohort study based in hospitals, where we enroll women mostly in their first trimester of pregnancy. We collect urine samples during each trimester, test them for multiple pesticide analytes, including glufosinate and glyphosate and dicamba and 2,4-D, and their primary metabolites. We then use standard epidemiological methods to assess whether the women with higher measured levels of herbicides in their urine during pregnancy were more likely to give birth to a newborn that displayed, some adverse impact from the prenatal herbicide exposures. It could be low birth rate, preterm delivery, breathing problems, or a range of birth defects. We also hope to be able to follow the mother-infant pairs and the infants and children that are enrolled in the Heartland study through age 16, using standard developmental assessments to really assess what impacts, if any, the prenatal herbicide exposures would have. The Heartland study goal is to enroll our 2000th mother-infant pair in early 2025, where if we can raise the money needed to expand the study, we're on a trajectory to do that. If we reach that goal, the Heartland study will at that time be the largest birth cohort study focused on herbicides ever conducted in the world. As part of, so obviously collecting urine samples and quantifying levels of herbicides and other pesticide analytes in the urine is an essential component of this kind of birth cohort study. We actually enrolled our first mother-infant pairs before COVID. We enrolled 18, and then COVID shut down clinical research in hospitals across the country for about 18 months. In that interim period, we decided to do a couple case control studies, drawing on data from previously conducted and completed birth cohort studies. We used the new mom to be cohort predominantly, which was a really elegant study on women during their first pregnancy. And the women enrolled in new mom to be came into the study between October of 2010 and September of 2013. So all of these urine samples are clearly before the Extend Dicamba tolerant soybean technology was present in the marketplace, and before there was any significant uptick in dicamba use. We also have tested at this point 91 samples from the women enrolled in the Heartland study after the COVID hiatus was over. These samples are mostly 2021 and 2022, 91 of them. And of course, all of these represent a time period after dicamba tolerant seeds were planted on something like 2/3 of the soybean acres in the state of Iowa. So all of those samples were tested for glyphosate and glufosinate and their primary metabolites. For those that aren't familiar with glufosinate, the common name is Liberty. It's a similar herbicide to glyphosate, and it's one whose use is rapidly increasing across the Midwest, because most weeds that are resistant to glyphosate are not yet resistant to glufosinate. So we have 152 samples that use this new method that HHRA developed in partnership with our analytical lab, the Center for Toxicological Research in Quebec, which is in effect the CDC of Canada. That new method detects 13 different pesticides and their metabolites, and including dicamba and 2,4-D. These are sort of preliminary rough calculations from the data that we have, and the findings are really quite striking. First of all, we detected 2,4-D above the limit of detection in 100% of the samples from both time periods. So 2,4-D is a ubiquitous contaminant now in the Midwest and most of the country, and almost everyone has it in their urine on a daily basis. But the frequency of dicamba residues in the urine of the women went up by about 50%, from 41% positive to about 65%. the change in the average or mean level of these herbicides was even more striking. As you can see, the mean level of 2,4-D went up about 45%, but the mean level of dicamba more than tripled. And, really surprising to us, probably by 2022, the average person in the Midwest was being exposed to more dicamba on a day-to-day basis than 2,4-D. There is there's been a significant rush of new science in the last year or two that is really having a substantial impact, I think, on how many people view the choices that farmers make in terms of how they control weeds and whether the current almost sole reliance on herbicides, and in particular glyphosate-based herbicides, in weed control on over 200 million of the 310 million cropland acres in the United States is really a sound management strategy. I follow this very closely, this area of science very closely, because I've served for 6 years as an expert witness in the Roundup non-Hodgkin lymphoma litigation. My responsibility among the experts working on the litigation is the sort of regulatory portfolio of issues and testing and what the tests show relative to guidelines that the EPA is supposed to abide by in evaluating the oncogenicity of pesticides. This paper in the upper left corner, I strongly urge you all to read it. It is probably the most significant paper on glyphosate and health risks, and in particular cancer and reproductive related risks, to come out in many years. It's the team of scientists are all either the scientific leaders within the National Cancer Institute of what's called the Agricultural Health Study, which is the largest, most sophisticated epidemiological study ever conducted, probably in the world, on the impacts of pesticides on various adverse health outcomes. Some of these co-authors are from the CDC and represent the analytical lab within the CDC that tested the glyphosate levels in the urine of the farmer applicators that were enrolled in this study. So basically what this team did is there were approximately 57,000 certified pesticide applicators enrolled in the Agricultural Health Study in 1993 to 1997. And those farmer applicators and some commercial applicators have been followed ever since. What this team of scientists did is they went in and they selected out a subset of the farmer applicators, about 1,600 of them. They contacted them, got a urine sample from them. They tested the urine at the CDC lab, Antonio Calafet's lab. So they had levels of glyphosate in the urine, and they used the same urine samples to test for markers of oxidative stress. They tested for 3 of the markers, and 2 of them showed a statistically significant association with glyphosate levels. This is extremely significant because it shows for the first time that the frequency of exposures and level of exposure to glyphosate is associated with markers of oxidative stress. Oxidative stress is clearly linked to damage to DNA, and damaged DNA is a precursor for cancer. So this is it's a very significant study, and it includes a remarkable sentence which says that their results in non-farmer controls or farmer controls that reported no use of glyphosate. So both of those groups of participants in this study would presumably reflect the average person, at least in rural America, that wasn't applying glyphosate. They had evidence in the control group and the no use of a glyphosate-based herbicide group of elevated levels of suggesting oxidative stress. So this is it's the first time in any study that I know of, and I've been tracking the literature very carefully, where presumably mostly dietary and water-based exposure to glyphosate has been associated with a marker of possible adverse effects. So it's a really important study. It came out last Wednesday. It's just getting recognized, and I can assure you it's going to have an enormous impact because of who wrote it. It's published in the official journal of the National Cancer Institute. Switching down to the bottom right corner, this is a paper that I did with my colleagues, Robin Musage, a terrific young scientist in Europe that's done more work on surfactants and herbicides and adverse health effects than really anybody. And William Sawyer, who is a toxicologist who has also worked on the Roundup non-Hodgkin's lymphoma litigation over the last 6 years. What Bill and Robin and I did was we collected all of the new genotoxicity assays carried out since the beginning of 2016. And this would be after the EPA completed its assessment of glyphosate oncogenicity and after the IARC working group had completed its assessment. So it's the new genotox data on both glyphosate and glyphosate-based formulations, and ask the question, well, what does the new science show? And the answer is there were 94 assays on either glyphosate or if a glyphosate-based herbicide reporting the results in a genotoxicity assay. And of them, 84 reported a positive result, or it's something like 72, 73%. And so and of the 72 or so studies that report a genotoxic response, probably close to half the authors said that the response was associated with or triggered by oxidative stress. So the new NIH CDC paper, coupled with this comprehensive review that Robin and Bill Sawyer and I did, really is going to strengthen the mechanistic explanation of why and how glyphosate-based herbicide use and exposures is likely contributing to cancer risk, and in particular, because of other data, the risk of non-Hodgkin's lymphoma. And this is so important because the EPA's classification decision on glyphosate herbicides that they're not likely to pose a cancer risk depends heavily on the Office of Pesticide Program's judgment that there is no valid genotoxicity data that suggests a possible mechanistic route through which exposures to glyphosate or a glyphosate-based herbicide can contribute to cancer. For several years now, many scientists have questioned, if not outright criticized, that judgment by the EPA. And this new paper that we've just published literally came out yesterday, I think it was. It explains why and how the EPA has tried to support that finding, but it's just not working. In the upper right corner, this is another paper that's just come out. It's an analysis that I did with the Dietary Risk Index system, which we're going to spend some time talking about in a minute. And it basically explains why the current way regulatory agencies and governments are trying to keep high-risk residues out of the food supply is not working, and is, in fact, is working very, very badly. Bottom left corner, Susan Kegley, Brian Baker, and I published this paper maybe a year ago. It marks I think the most sophisticated data-driven analysis of the residues and risk in fruits and vegetables grown in conventional systems compared to residues and risks in the organically produced fruits and vegetables. We go about it in a couple of different ways, and I think it's a paper that's added a lot to the literature. So these are studies that have just recently come out, and there's a bunch of new science coming. We're going to be publishing a biomonitoring paper with our Dicamba and 2,4-D levels, and the significance of our Dicamba data is this. We can think of no plausible explanation of how 2/3 of the women in a heavily corn soybean farmed part of the Midwest could have dicamba in their urine other than through inhalation exposure. Many of these women, in fact, most of the women came from suburban and urban areas around Indianapolis. They're not sitting out in the middle of corn soybean fields in a rural part of the state of Indiana. So it's possible that drinking water and beverage-based exposures contributed some. Obviously, we know there's a lot of lawn care use with dicamba, which explains why 30 or 40% of the women tested positive before XtendiMax technology came on the market in 2017. But we think the current evidence is pointing to inhalation exposure from the dicamba that first volatilizes, moves up into the atmosphere, and then moves en masse, and including it moves over the city of Indianapolis and over the city of Bloomington and Chicago and Minnesota, and on and on with the major urban centers through the Midwest. We'll be doing a lot more work and more publishing on dicamba and 2,4-D levels in the urine of pregnant women. There's more papers will be coming on residues and risks in organic food. Our Heartland Study protocol and methods paper will be published, and a very important study, the global glyphosate study that's been underway at the Ramazzini Institute since 2018. The results of that will come out in 2023, and that includes a 2-year rat oncogenicity feeding study, a 2-generation rat reproduction study, and a full battery of genotoxicity assays. The critical difference in the global glyphosate study compared to all other toxicological research done on glyphosate and GBHs is that the Ramazzini team has used relatively low doses. The dosing range is in fact very similar to the regulator's assessment of a chronic reference dose. So basically, the dosing range is about 100-fold lower than any other previously conducted study. So if the global glyphosate study does identify any adverse impacts at these much lower dose levels, it will put substantial pressure on regulatory agencies around the world to lower the chronic reference dose. Since I'm talking to a lot of folks that are involved with and supportive of organic farming, I want to spend some time talking about how our understanding and knowledge of the difference in pesticide residues and risk in organic versus conventional food has evolved. I've had the privilege of, for about 25 years, working periodically with the Consumers Union, Consumer Reports folks, and my analytical work from the early versions of the Dietary Risk Index was featured back in March 1999 in How Safe Is Our Produce, and then about 16 years later we did a fresh cut of it, and these two investigative stories in Consumers Report really did a lot to highlight for the general public the fact that if you wanted to avoid pesticide residues in food, and in particular fruits and vegetables, the best and most reliable strategy was to buy certified organic brands. So over the years, we developed what's now called the Dietary Risk Index system in order to compare the frequency and levels of residues and risk of pesticides in food. The schematic in the left side, that is the Dietary Risk Index system. There's 30 years of work invested in building this. There's actually, on our website, there's a section on the history of the DRI. The seed of the DRI was planted back in the early 1980s when I was the ED of the Board on Agriculture in the National Academy of Sciences, and we were funded by EPA to do two important reports on pesticide residues and risks, and we had to develop a new method to quantify risk. So we started on the path that has led to the modern DRI. One of the important applications of the DRI is that we can compare residues and risks in conventional apples to residues and risk in organic apples. We've done that. We've been able to do that for several years, and now I'm going to walk you through some of the new technology that's available for anybody to explore these differences. In the last year or so, we've been working with a few certifiers and the ACA to develop what's called OrgTracker, and basically it's an enhanced module within the DRI to track the pesticide residues and risk levels in organic food based on the residue results collected by certifiers in the course of adhering to their requirement to test food from 5% of their certified entities for residues every year. I'm going to see if I can pull off a switch here to so you're seeing now I'll walk you through the whole steps here. So this is the homepage of the Heartland Health Research Alliance, which I serve as the ED of, and which is carrying out the Heartland Study here. You can see our sort of the scope of our work, and then there's ways to get at different sets of information and data. So under our projects, if you go to the Dietary Risk Index and to About the DRI, you land up on this page, and this page was designed for somebody that never heard about the Dietary Risk Index, but is interested in using it and trying to understand the data that it's producing and where it came from. This index of DRI pages, were all developed again for people that are new to the DRI. We explain basically how it's used, the history of it. There's a longer version of the schematic. Here's two published methodology papers, and then kind of step-by-step guide and how to look up different information, and then a whole series of different ways to interpret the values that come out of this system. It probably would take somebody 45 minutes to an hour to read all this, but if you do, you really understand what's going on here. So I'm now in the dietary risk index. This is all live, it's interactive, it's free. And there's 1, 2, 3, 4, 5, 6 different modules. Each one of these is a module. And within each of the modules, there's anywhere between a few thousand to 20 or 30,000 tables, unique tables that can be generated. We haven't done the math, but if you count up the unique tables across all the selection criteria that the DRI system now generates, it's over 1 million. So let's just start with the most simple. This is by food. So, and I'm in the part of the DRI that's based on the US Pesticide Data Program testing. There's also up here in the corner, if I can figure out a way to, you can see switch to the UK FSA data. So we've logged into the DRI system all of the UK Food Standards Agency pesticide data, and it really turns the DRI into a system assessing pesticide residues of risk in the global food supply. Okay, so there's all these different foods that the PDP has tested. I've selected apples and all samples, so that's domestic and imported. I've selected all market claims, which are just organic and conventional equals all samples not labeled organic. And then these are all the years for which we have data. All of the tables go back, or most of them, to 1992. So we've got 24 years of data in this. So this is the first basic results table. These are all the pesticides that were found in apples in 2016. There were 531 samples, of most of them tested, but for a few others, there was a different number. For example, and one naphthol had 442. These are the number of positives, the percent positive, the mean residue level. This is the chronic reference concentration. This is the core parameter in the dietary risk index, which is used to rank the dietary risk from thiabendazole residues in apples in 2016. This chronic reference concentration is driven by the chronic reference dose as set by the EPA. So it's your basic measure of chronic toxicity. There's 2 different forms of the DRI. One we call the DRI mean, or M, and the other is the food supply DRI. The DRI mean is based on just the 334 samples of apples that contain thiabendazole. This is the DRI mean represents the risks from the average risk from thiabendazole in those 334 samples. The FSDRI is the risk from thiabendazole in all 531 samples. So in almost all cases, the FSDRI is smaller than the DRI mean, and the smaller the percent positive, the bigger the difference becomes. This last column is the percentage of, if you add up all the pesticides in apples across the FSDRI, this column ranks them by the share of each pesticide. So basically, almost 1/3 of the risk was accounted for by the 334 positives of thiabendazole. One of the things as you look across foods is you'll see that just a few pesticides for almost all foods account for 90% or more of the risk. So let's say you're not interested in apples, but you are interested in grapes. So you just pick grapes, and here's the grape table. And here, cyprodenil accounts for 33%. Oh, so let's say you want to check out the domestically grown grapes. So we just pick domestic grapes. And now these are the pesticides and risk found in the 335 samples of domestically grown grapes. And in this case, the aggregate FSDRI is 0.03. So at the bottom of all of these tables, there's basically summary data. So there were 1,700 residues found, an average of almost 5 residues per sample. There were 46 different pesticide analytes found, and here's the DRI mean, FSDRI. So you'd say, oh, that's domestic. Well, let's look at imported. So we can go to imported, and there were 350 samples of imported grapes. And you could see down here that there were about the same number of samples and much lower DRI mean, or actually wasn't lower, but similar relationship here. So you can go through.
Dr. Benbrook, sorry to interrupt. So this is for conventional grapes. What about organic grapes? Is there?
We're going to go there right now.
Oh, gotcha. Okay, sorry, I was.
So this is the current module that compares conventional organic. And so we're going to do apples in 2016. Okay. And so if you go down, there's 3 tables that summarize these data. So this is your summary table. There were 481 conventional samples, 16 organic samples. So there was 30 samples of conventional apples for every 1 sample of organic, which is way less than what they should be, because the PDP program guidelines call for sampling conventional and organic food roughly in relation to the market share of the 2 production systems. Here's the residues found. The average number of residues per sample, again, almost 4.5 per conventional sample, and 1/2 for organic. And look at the huge difference in risk levels, almost 500-fold in the case of DRI mean and 400-fold in the case of the FSDRI. So you might wonder, well, what residues were in the organic samples? So that's what table number 2 shows. There were 16 samples that were tested for diphenylamine. 4 contained residues. So of the 16 samples that were tested, there were 4 diphenylamine residues, 1 spinosad A, and 3 thiobendazole. So this gives your percent positives and the risk levels. And look how tiny they are compared to the conventional. So diphenylamine is in apples because it's a post-harvest fungicide. Spinosad A is in apples because there is a organically approved formulation of spinosad that's used by some organic apple farmers, and thiobendazole is in organic apples because it is not possible in some packing houses to clean the thiobendazole residues out of the tanks that the apples are washed in. And so when a packing house shifts from running conventional fruit to organic fruit, there's often residues of post-harvest fungicides. And this is a very important factor when talking about the overall presence of pesticides in organic food. So this last table 3 gives you information about the 3 residues that were found in organic apples in 2016. 2 of them were post-harvest fungicides. Number of positives. Now, here's the tolerance level. So according to the NOP rules, any residue over 5% of the tolerance cannot be sold as organic. So this calculates the 5% of tolerances. This field, residues over threshold, we haven't got the system to report this yet, but there's none in the case of organic apples. And then this concept of inadvertent residues is another one that we've used in our peer-reviewed papers. And it basically says if a residue is present in an organic apple at 1/10 or less of the mean residue level for the same pesticide in conventional apples, then it's an inadvertent residue. And by that, we mean if a residue is 1/10 of the mean of the residues in conventional, that residue in organic was not a result of a direct application on the crop, because an application resulting in such a low residue level would not be efficacious. So it's from drift or cross-contamination or contamination in the packing house, et cetera. And then this last table just conveys all of the pesticides in conventional apples. So now I'm gonna, whoops, wrong one here. Sorry. So remember I mentioned OrgTracker. We're building this in conjunction with some certifiers. This is a mock-up of what the landing page is going to look like. So there'll be data from different certifiers that go into this master database. We'll combine the results, hiding both the identity of the certifier and the grower that the residues came from. And we'll do the standard output tables like we've shown. And we'll also do it based on individual samples. I didn't get a chance to talk to you about that other important module within the DRI. And then we'll design several summary tables that are intended to answer questions that certifiers have on their minds about, when you get back, when a certifier gets back 120 samples of organic food from their certified entities with levels of various pesticides, they have no way to identify which of those residues is something that they and the growers and the public should be concerned about versus the inadvertent or levels that are very small and really reflect legacy use of pesticides. And then we'll build in a number of ways to explain to people using the system how it's to be used, et cetera. There'll be a private password protected part of OrgTracker that only certifiers can get into, and a second level of password protected content where CCUF, for example, will be able to get tables based on its data that shows the relative ranking of risks of the samples that it took, paid for, and need to figure out what to do about. So OrgTracker will produce tables like this that we just went over. And so this is what we hope the certifying community will come to see as the benefit of a data-based analytical system like the DRI to figure out what to do with this investment that the organic community is making in residue testing. So obviously, this system is going to be very reliable in identifying the relatively few samples of organic food that contains a residue posing a risk level that we don't want to see anybody consuming in their food. The results are going to clearly show where the high-risk samples are coming from. I can tell you, for example, if you look at peaches or apples or grapes or berries, a lot of the Pacific Northwest and California production is pretty clean, but production in Michigan, North Carolina, Georgia, other states where it's more humid and wetter tends to have a lot more pest pressure and hence more residues and risk in conventional food and more residues and risk in organic food. It'll also help certifiers recognize and understand which samples from what growers and what regions are more regularly associated with a residue. So what's in OrgTracker for the NOP and the broader organic community and organic food companies and retailers that want to provide their customers with a choice of essentially pesticide risk-free food? Well, if OrgTracker is used and then certifiers in the NOP rely on the data to identify the very few high-risk organic samples that are in the food supply and then go after them in more frequent testing and enforcement actions. And in the worst case, dropping their organic certification. That's going to clean up the organic food supply chain pretty rapidly. I would say 5, 6, 7 years, this system should reduce the levels of pesticide risk in organic food from the very low level now to, you know, a 10th or even 100th-fold lower, and so essentially eliminating. And if we're able to do OrgTracker and on an annual basis have certifiers, most of the certifiers in the country submit their residue testing results to OrgTracker, that is quickly going to compile the largest data set in the world on pesticide residues and risk in organic food, and it will allow scientists to do a much better job in quantifying and comparing the risks. Well, sorry about the whirlwind tour there. I know it's a lot to get through, but really glad to have this chance to visit with you, and let's do some questions.
Yeah, that was fascinating, Dr. Benbrook, just incredible. As far as some of the research papers that you mentioned at the beginning of your presentation, Danny asked, there was one paper that said that it is coming soon. Do you know when it will be published? Do you have any idea on that?
The 4 papers in the first slide, they are all published now. The papers that are coming soon, I'm going to guess our Dicamba biomonitoring paper might be out as early as March and certainly by April. The methods paper for the Heartland study will be out probably in March. And the Ramazzini Institute papers, there'll probably be 3 or 4 published in 2023. And I suspect the first one will come out in March, but the most highly anticipated one on the results of the rat oncogenicity study, I'm going to guess that that won't come out until the fall.
Okay, that's good to know. Thank you for clarifying that. There was a few comments when you were demonstrating the DRI tool on how powerful it is and how incredible the tool is. So thank you for demonstrating and sharing that with us today. Raphael said, are there any other results available than the ones from US? And you mentioned that there were some from UK. Are there any others besides the US and UK, I guess?
The DRI contains the 2 most rigorous and highest quality sets of residue data in the world. The data collected by the USDA's Pesticide Data Program and the data collected by the UK Food Standards Agency. The UK Food Standards Agency uses a different set of criteria to select samples. And they also test far fewer samples per year of a given food, but far more foods. The UK imports 60% of its food supply. So there is a heavy focus in the UK FSA testing on imported foods. And we're talking everything from beer and spices and tropical fruits to cheeseburgers and multi-ingredient frozen meals, etc. So the UK FSA data provides a very broad assessment of residues in the food supply. And like the PDP, you know, they pick 5 to 10%, sometimes a little bit more than 10% of their samples from the organic labeled product in supply chains. So by combining the PDP data, which really is heavily focused on fresh fruits and vegetables and other foods that play an important role in the diet of pregnant women, infants, and children, by statute, that's what the PDP program was initially designed to do in the early 90s and still does to this day. So we've integrated these 2 data sets now and really have the capability of assessing the distribution of residues and risks in much of the global food supply. For example, we have the UK FSA has some data on bad actor foods like frozen cherries from Turkey, which can have some really nasty residues in it. So we are getting closer to having the capacity to really track residues in a significant portion of the global food supply.
That's amazing. That's really incredible. Thank you for all the hard work that you've put into that, Dr. Benbrook, and also sharing with us. Dr. Audrey Tran-Lam, do you want to unmute yourself and ask you a question? I see you have your hand raised via Zoom.
Yes, I would. Thanks so much, Olga. Dr. Benbrook, thank you so much as always. I always really enjoy hearing you talk and share about all of this information. I have so many things to comment on, but since I have to hop off really quickly, I just wanted to clarify my own understanding of some of what you were describing with the oxidative stress in some of the recently published papers earlier. In thinking about how I want to be explaining the meaningfulness of these new papers, I think what I was hearing you say was that these papers on oxidative stress have really been the biomechanical, the mechanistic missing link so far in putting these pieces together on the potential harms of population exposure to pesticide residues. Do I have that right? Like, were you saying that this is these are so impactful because they're sort of representing that missing link that we now are starting to have?
Audrey, great question. So the three pillars of assessing the oncogenicity of a chemical and certainly including pesticides is the animal studies. So these would be the 2-year rat and mouse bioassays, the epidemiological studies in exposed humans, if any, and then the mechanistic or genotoxicity data, which covers whether there are known to be a biological mechanism through which exposure to the chemical of interest can damage DNA or alter gene expression in ways that increase the risk of cancer. Now, all of us are experience and generate a degree of oxidative stress in our bodies every second we're alive. Oxidative stress is a normal part of cell respiration. It's something that our bodies deal with successfully for most of us throughout our lives. Our endogenous production of antioxidants, i.e. what our bodies produce, is highest through our 20s and 30s and then starts to decline steadily as we age, which is why it's even more important for the elderly to consume their recommended numbers of fresh fruits and vegetables, which are typically much higher in antioxidants than any other part of the food supply. So what this new National Cancer Institute CDC study is reporting that the farmer applicators that used sprayed a lot of Roundup or glyphosate-based herbicide, the markers of oxidative stress among those individuals was elevated above the levels in the other farmers or non-applicator controls in the study. So we all have oxidative stress every minute that we're alive throughout our lives. But the question is, do our bodies generate enough endogenous antioxidants, and do we supplement the levels of antioxidants circulating in our blood sufficiently to keep the so-called free radicals from bouncing around inside our cells and damaging DNA? Because when oxidative stress gets inside, oxidative stress can lead to a breakdown in cell walls where chemicals in your blood, in your body, can get into the inside of cells, and that's where, of course, a genotoxic impact can occur that triggers the cancerous or other abnormal growth. So the oxidative stress is a huge topic in the whole area of pesticide risk assessment, because there are a lot of different pesticides that stimulate oxidative stress. But this new study by NIH and CDC is really the first I know of that really, in a very rigorous study in a farmer applicator population, demonstrates an elevated set of markers in the urine of these individuals indicative of higher oxidative stress.
Audrey says that's very helpful. Thanks so much, Dr. Benbrook. Okay. Thank you, Audrey, for being here today. Are there any other questions, guys, for those of you who are on the call? I have a question, Dr. Benbrook. I don't know if that's something that you know, there is much research on, but I've heard that over time the antioxidant level in our conventional foods are decreasing with time. Has there been any research to reflect that type of conclusion? And if so, you know, can you share more on that?
Yeah, great question, Olga. There is a huge body of literature that documents somewhere between 15 and 25% lower total antioxidant capacity in conventionally grown fruits and vegetables compared to organic. The explanation for that is predominantly the dilution effect, the effect of high nitrogen systems growing bigger apples, pears, cucumbers, heads of broccoli, which are primarily composed of water and carbohydrates. When a plant has lots of water and lots of nitrogen, even more than it needs, the only way it can use it is to just grow, is to convert the extra nitrogen and water into carbohydrates. So that's why you bite into a juicy big apple. It's so sweet. Well, all of that, the carbohydrates that lead to that sweetness, they're adding to the mass, the volume of the apple, and diluting the concentration of antioxidants in the apple. So that is a widely accepted finding across, I mean, hundreds of comparison studies done around the world. Now, there have been some clinical studies in human populations where association has been reported between ingesting predominantly organic foods and in particular organic fruits and vegetables and dairy and reduced markers of oxidative stress and higher antioxidant intake. So this is there have been some studies that point this out, but to do a rigorous clinical study over a long enough period to pick up the impact of higher antioxidant intake from consuming predominantly organic fruits and vegetables would cost tens or $100 million, because you need to follow people for at least several years, and it'd take a lot of people. You'd have to have a lot of knowledge and control over their diet. So that's why there really haven't been any conclusive studies on that topic.
But based on the research that you just mentioned, it's safe to say that organic foods are not only more more full with antioxidants and better food qualities and less pesticides versus the conventional counterparts. Correct.
Exactly. Some of the research I did back in the days when I was the chief scientist at the Organic Center, we took for about 125 different common foods, we calculated the total antioxidant capacity per calorie in a serving of food. And one of the remarkable things that we found was that a consumer can pick an additional serving of organic fruit or vegetables and get about a 25% bump in their total antioxidant capacity, which is kind of like a whole nother serving of food without any additional calories. So for across the US population, the capacity of organic farming systems to produce more nutrient-dense food that delivers significantly more human nutrition per calorie consumed is a critical parameter, and one that the Heartland Health Research Alliance will be talking about and featuring in our forthcoming comments to the FDA on the open rulemaking on how to define healthy food, and how the FDA might structure a front of label food labeling system in the future, designed to give consumers reliable information about how healthy a given food product is.
Wow, that's fascinating to say the least. When you say all of this, all I can think of are the school meals in our school systems. And how, these young kids, I personally am a mom with young children, one of which is in kindergarten this year, and so just balancing that whole giving lunches to a child, and not always doing that, and allowing them to eat at school, and full well knowing that none of the school fruits and vegetables that they're getting are organic. And I feel like it's so important to put that at the forefront of FDA and USDA, and let them know how critical it is to introduce those types of foods for these young developing children.
Yes. Yeah, I'm often asked why the demand for organic food in the United States hasn't gone up more significantly, particularly for fresh fruits and vegetables that account for probably 90% of the pesticide dietary exposure and risk. And I think, after really pondering this question for 20 years, I think it's the mixed messages that come from the food industry and the government about whether these food nutritional quality, food safety benefits of organic are real or just marketing hype. I really feel that the efforts of interests in American agriculture that see the growth in the organic sector as a threat to their market share and income streams, they've been successful in casting enough doubt that a significant segment of the population is not convinced that it would make a big difference in their health, especially in the years when couples are trying to get pregnant and are raising young children. I mean, that's the time to eat really nutrient-dense food, and food that has minimal pesticide residues in it. But I hope at some point the science will justify a clear message to consumers. And I think that's when we'll start to see growth in certainly organic fruits and vegetables increase again.
Dr. Art Dunham had a comment in the chat box, and he said that noticed that neither dicamba or glyphosate levels were looked for in your apple example in the DRI system. As a veterinarian, I get frustrated with our look at antimicrobial resistance, looking at antimicrobials used on livestock without looking at glyphosate level as well.
Yeah, it's a good comment. The US PDP program does not test food for glyphosate. They only did one special survey of soybeans about 10 years ago. Glyphosate requires a separate method that is costly, about $125 to $150 per sample, which is one of the reasons the Heartland study is so expensive to carry out. And the USDA has never tested for dicamba. We certainly wouldn't expect much glyphosate or dicamba to show up in fruits and vegetables because they're not applied directly on the crop. But there certainly could be some drift. Glyphosate is in the urine of most of us on this call, except for the people that are really rigorous in choosing only organic food, and particular grain products. from pre-harvest desiccation uses in wheat, oats, barley, and edible beans. That's why glyphosate's in the diet and why it's in people, the general public.
Yeah, thank you for sharing that additional aspect of the testing of glyphosate and or not testing. There is a different resource, the Environmental Working Group, that puts out the Dirty Dozen and the Clean 15 list that shows the Dirty Dozen, which is like the worst possible offenders, like the fruits and vegetables that you mentioned, like apples and strawberries and things of that nature. So I personally try to adhere to that as much as I possibly can. And I try to share that with others that are interested to buy organic. And ultimately, like if an avocado doesn't have as many traces of pesticides on it and I don't have to buy it organic, I'll skip it that way. So just knowing and having those tools and resources available to you, I think are important, just to kind of guide yourself through, your purchases when choosing organic foods and when staying within a budget.
Well, thanks everyone for having me, and I look forward to visiting with you again down the road.
Yes, thank you, Dr. Benbrook. And for everyone that was on the call, I will be sending a link to the recording that will be posted on our YouTube channel, as well as a quick survey that we'll be using for grant reporting purposes. So please take the 5 minutes to share your thoughts, and that would be greatly appreciated. And Dr. Benbrook, this was incredible. Thank you so much from the bottom of my heart, and I'm sure everybody else enjoyed it just as much. Fascinating. I look forward to checking out the DRI tool myself moving forward, now that I know how to use it. So thank you for the demonstration.
Okay, bye-bye.
Everyone, have a great day.